So good morning students. Today I'll continue with external genitalia in female and then continue one more subtopic, that is spermatogenesis. So yesterday I was explaining about external genitalia in female. So here so the first what has been completed is the mons pubis, the predominant, the elevated portion within the external genitalia and its characteristics. Later on, about the labia majora and minora. So labia majora is the outer boundary, whereas the outer fold of a skin which is much larger, whereas labia minora is the inner fold of a skin which is much smaller. And today I'll explain about the labia minora. So labia minora forms the inner skin. So this portion was the outer skin fold and this portion was the inner skin fold, and this will be the mons pubis, and this structure is the clitoris. So this inner fold of a skin is called as labia minora. Now, schematically, as I told yesterday, the mons pubis, the outer fold of a skin, and then the inner fold of the skin. This inner fold of the skin itself is called the labia minora. Now, anteriorly, you can tell, or in the front. So, anteriorly, here it forms a junction. Okay, at this junction, there is a structure called clitoris, about which I am going to explain shortly. So, this is about the inner fold of the skin. Now, the labia minora here, this is the fold of the skin here. Now, the space that comes in between these two folds of the skin is called as vestibule, okay, or vestibular region it is called. So, that was about the labia minora, the inner fold of skin. So, anteriorly or at the junction, it is called as structure forming called as clitoris. Now, posteriorly, the labia minora posteriorly will fuse at forecheek. The distance between forecheek and the anus will be called the perineum, but that is, however, not there. Yes, so labia minora. So labia minora anteriorly have a structure I told, and that is called clitoris. So clitoris is a very much it's a homologous to penis in male. Homologous to penis in male. Now there are some similarities with that of the penis. Also there are some differences. So first of all, where this clitoris is located. So anteriorly or in the front, you can tell anterior junction of labia minora. So here the labia minora anteriorly, this finger-like projection is located. So anterior junction of the labia minora. Then what kind it is? It is finger-like in structure. It is finger-like in structure, and it has an erectile tissue. Okay, it is erect. It consists of erectile tissue and is a sensitive region. So it consists of erectile tissue. The name of the erectile tissue is corpora cavernosa, and it is a sensitive portion. So then, how does this differ from the penis of male? Okay, so in what ways it differs from the penis of the male? Compared to the penis here, it is reduced in size. First point, compared to penis, it is reduced in size. Then second point is that it does not have any passage, but the penis has the passage. So there is no passage present. So that makes clitoris different from penis, even though they're homologous structures or homologous structures they are when compared to male and male human and female human. Okay, so anterior to it, that is above the clitoris, what is going to be situated? So again the same diagram, mons pubis, above the clitoris will be the labia majora, then above it again will be mons pubis. So this was about what is called a structure that is present in the anterior junction of the labia minora called the clitoris. Then coming to a membrane. So the labia minora has structure clitoris, which also is associated with the foreskin, which also has the prepuce also. So here, space here in between the labia minora, this is called as vestibular region. That is a vestibulum or vestibular region. Okay. So here, this will be the vestibular part of the labia minora. So in the vestibular structure, what are supposed to be present? So there are two openings in the which is there in the vestibular region of the external genitalia. So two openings in the vulva, two openings in the external genitalia. So first opening which is present above, and that is called urethral meatus. And the second opening which is present below is vaginal orifice. Is the vaginal orifice. It's also called as urethral orifice. Urethral orifice. So there are two openings. Now, purpose of urethral meatus will be the release of urine. So urine is going to be released through urethral meatus. So labia minora. So two openings on above will be the urethral meatus and below will be the vaginal orifice. ಅದು ನಾನು ಆ ಕಡೆ ಹೋಗ್ತೀನಿ ಅದು ನನ್ನ ಕಡೆನೇ ಮಾಡ್ರಿ ಗೊತ್ತಾಗ್ತದೆ ನನಗೆ ಈ ಕಡೆ ಬರಕ್ಕೆ ಯಾಕಂದ್ರೆ ಔಟ್ ಆಫ್ ಸ್ಕ್ರೀನ್ So two openings are there. One is for the let out of the urine, let down of the urine. Another is for one is for the ejection of urine. Another is for So here it can be the ovum, it can be the menstrual discharge, or also in case of pregnant woman can be the fully developed fetus. Fully developed fetus. It can be the fully developed fetus. So then of course what I have completed is the majora and the mons pubis. So these are the two openings and their functions. See, the opening of the vagina will be partially covered with a membrane here. This membrane has a term and that's called hymen. So partially covers the vaginal opening, also called vaginal orifice. Opening is called orifice. So now I'll be explaining about the hymen. So hymen is a membrane that covers partially, covers what? The vaginal opening or the vaginal opening. So that is very important that the coverage is partial covering. It's called the partial covering. Understand? So partial covering is there in due to this structure. Now, this particular membrane that covers partially, this can rupture. There are different reasons why it can rupture. The reasons is it can be due to it can break in the say first intercourse. It can break, rupture due to use of say tampons, okay, which are used. These are the sanitary tampons. The next will be, it can be due to sports activities, that to regular sports activities like horse riding or like other sports. Sports. It can be due to sudden jolt. Okay. Horse riding, cycling, etc. Cycling, etc. So in the earlier days, it was been used as it's used to determine the virginity of the female. But now, since it is known that it can also rupture due to various causes as mentioned here, can rupture due to various causes as mentioned here, and also certain women can by birth itself will not have the hymen, and certain women can have persistent hymen even after various activities. It is not going to rupture. So therefore hymen cannot be a reliable method to determine the virginity of female. So this completes the next part of the external genitalia called the one. Now here there are some accessory glands present here in the female reproductive system. The glands present in the female external genitalia, especially not the entire reproductive system, but confined to the external genitalia of female. So here, what will be there is the greater vestibular gland, and one more is lesser vestibular gland. These two are actually accessory glands. Accessory glands. Greater vestibular gland, for example, will be Bartheline gland, and the lesser will be the skin gland. So this is not there in series, so I'm going to brief it. So Bartheline gland is a racemose gland which appears like a bunch of grapes, and it is similar to the bulbourethral gland. Okay, so it is going to secrete an alkaline fluid and help in lubrication. And the ducts of this particular gland is going to open on either side of the vaginal orifice. Whereas lesser vestibular gland are skin glands, which are still comparable to, shares homology with the male prostate glands. And here what happens is it is also going to, its ducts are opening on either side of urethral meatus or urethral orifice. So that means the two openings that are present in the vestibular gland will have ducts of these glands opened up. So therefore, this will be about the accessory glands present in the female reproductive system. So next, I'll continue with gametogenesis. So next will be spermatogenesis. So this is an NCERT diagram for the spermatogenesis as to how the sperms are. So this is so spermatogenesis means what? It will arise from another term called gametogenesis. What is gametogenesis? Is production of gametes. Production of gametes is called gametogenesis. And what are gametes? You all know the gametes are nothing but the sex cells. Okay, so gametes are produced through the phenomena that is going inside the reproductive system, and that is gametogenesis. So it's not only applied for human beings, it's a very general term as to what is gametogenesis. Now, that is general term applied for other animals also. So now gametogenesis can be two types. Further making it more organized will be the spermatogenesis and oogenesis. Spermatogenesis and oogenesis. A spermatogenesis is what? Again, it's easy. It is nothing but production of sperms, and here production of ovum. And there is another term, production ovum, or also you can tell as eggs, production of ovum or eggs. Now, where do spermatogenesis occurs? Where do this gametogenesis occurs in generally means it occurs in a particular organs that are present in reproductive system. And those organs where this gametogenesis occurs can be called as gonads. Okay. Now, what are gonads? Gonads are primary sex organs. Gonads are the primary sex organs. Okay. So the primary sex organs themselves can be called as the gonads. Now, spermatogenesis is a type of gametogenesis. Now, what is the gonad in which spermatogenesis occurs? Will be what? Testis. So, testis is a gonad. It is an example of gonad. And where do oogenesis occur? Oogenesis is going to occur again in an organ, ovary. Is going to occur in an organ, ovary. So here, these two, therefore, are examples of gonads. They're examples of gonads. Now again, I told that this testis and ovary are examples of primary sex organs. Now, primary sex organ means what? Already finished, probably finished in the earlier classes. So primary sex organs can be called so for two reasons. Because of two reasons, they're called as primary sex organs. They should be able to produce the sex cells or the gametes, which are nothing but the sperm and ovum or egg. Ovum also called as egg. Now, second is they also should produce specific hormones, sex-related hormones. It can be the male sex hormones or the female sex hormones. For these two reasons, now these will be called as the primary sex organs, the gonads, that is the testis and ovary. So this completes the definition of spermatogenesis. Production of sperms is called spermatogenesis. Where do these occur? Location, where exactly the sperms are produced. Very generally speaking, the sperms will be produced, as I told already, in an organ called testis. Now, testis, cross-section of testis has already been taught for you. So, testis itself is further divided into compartments. So, many compartments it has. In each compartment, 1 to 3 coiled structures. This present is seminiferous tubules. They call as seminiferous tubules. Now, this is where exactly spermatogenesis is going to occur. It means that the site where the sperms are produced within the testis itself. So, exactly within the testis where they are formed will be within a compartment where 1 to 3, what is called 1 to 3 seminiferous tubules will be there in each compartment. That is the site where the sperms will be exactly produced, or meiosis occurs and sperms are formed. Now, how is cell division related to spermatogenesis? So, type of the cell division. So, type of the cell division that results in production of sperms, and that cell division is meiosis, because sperms and ovum both are haploid, and the cell division that gives rise to the haploid cells is nothing but the meiosis. So, cell division that leads to, that is the basis of both spermatogenesis and oogenesis, is meiosis. Now we should therefore know that due to meiosis only, what occurs is spermatogenesis occurs as well as oogenesis occurs. So therefore, cell division that forms the basis for spermatogenesis, and even oogenesis also is meiosis. Now, are all cells capable of undergoing meiosis? No. There are only certain cells that are capable of undergoing meiosis, and that's called meiocyte. Now, that is called as meiocytes. What is a meiocyte? Cells capable of undergoing meiosis. Cells capable of undergoing meiosis is called as the meiocyte. So this was a kind of introductory, this is the introduction for spermatogenesis. Now remember, also meiosis produces haploid. Now also you should know that sperms are haploid. Sperms are haploid. How you can see, the total number of chromosomes in human will be 46 in the somatic cell or the body cells. Whereas in the sex cells like sperms and ovum, or in the already differentiated germ cells which are formed, the total number of chromosome is reduced to half here. Instead of 46, it is what? 23. So here itself, what here itself so here itself what happens is that half number of chromosomes the chromosome number is reduced to half, means these cells, now these cells is half number of chromosomes, these all cells will be having full number, that is 2n, and this is n. So diploid and haploid. So sperms are haploid. Now, from which cells do sperms start forming? There should be some meiocyte, the cell that is capable of undergoing meiosis, that should be present. Now, early in the embryonic stages, there are cells called as primordial germ cells that are capable of undergoing mitosis and then producing the cells like spermatogonia that are able to form the sperms. Now, even in the embryonic stages, these primordial germ cells won't be located in a particular region. They tend to migrate from early gastrulation to back into to the yolk sac, and migrate from the gastrulated stages to the yolk sac, and then to the region where the gonads are produced. And once gonads are produced, they start differentiating and then start forming the what is called the sperms, or even case ovum. So therefore, first cell, what is given here is spermatogonia. Spermatogonia are the cells, it is called gamete mother cell. Gamete mother cell that undergoes meiosis. That will undergo meiosis. Now, from where the spermatogonia has come, earlier part in the embryonic stages, there are some precursor cells called primordial germ cell from where the spermatogonia will arise. Now, one more thing, what has to be known as, the sperms do not start producing in the embryo stage or fetal stage. It cannot be produced there. But which is the exact period from where the spermatogenesis begins? So production of sperms starts in the puberty, and therefore who has the role for this is of course hormonally controlled. Hormones play a key role for this transition from the juvenile stage to reproductive stages. Okay, So therefore, here what happens, spermatogenesis will begin in the puberty, and later what happens is it's due to hormonal influence. Let me mention those hormones like gonadotropin releasing hormone produced by hypothalamus, which will stimulate the pituitary to produce the FSH and LH, and these two are called gonadotropins, and those are the one that brings about the changes that occur within the puberty. Therefore, that is how spermatogenesis begins. Now, compared to oogenesis, spermatogenesis almost occurs daily in the male, but oogenesis occurs only once. But however, to complete spermatogenesis, 74 days are taken. So, first, starting with the first cell, spermatogonia. Spermatogonia will undergo not meiosis. Before it could undergo meiosis, it is going to multiply. So it undergoes mitosis. So this will undergo mitosis. What is a mitotic division? You all know already. Mitosis is nothing but a cell division where the chromosome number of a daughter cell are same like that of the chromosome number of the parent cell. So when the chromosome number is equal, that kind of a division is called as equational division. So therefore, mitosis is an equational division, and the cell that is capable of undergoing that will be nothing but spermatogonia. Spermatogonia is a diploid cell. Next point regarding the spermatogonia is it is a diploid cell. Which cell is called as a diploid cell? The cell that has complete number of chromosome, they are called as a diploid cell. They are basically majority of the somatic cells are diploid. Now here in human beings, what is a diploid condition? Diploid condition is referred by 2n, which refers to the total number of chromosome of how much? 46. So total 46 chromosome is represented as 2n, which is also called as a diploid cell. Now, next point, what has to be known as, since this spermatogonia forms sperms, okay, it has to undergo mitosis and then start producing daughter cells. All the daughter cells are same or are they different? They're different actually. So what happens, let me see, this is a diploid. Let me say this is a diploid spermatogonia. This is a diploid spermatogonia. This is undergoing mitosis. This is undergoing mitosis and therefore forms two daughter cells. Then how will be the chromosome number of daughter cells? Equational, because it's undergoing mitosis. So in mitosis what happens? Two daughter cells are formed here, but the chromosome number remains same. 2n represents the chromosome number, and which is equal to total of 46 in human. So spermatogonia undergoes mitosis, produce two daughter cells. The question is whether all the daughter cells are same or they are different. They're actually different. There are two different groups of cells formed after those spermatogonias will undergo mitosis. Two different groups of cells will be formed. One is called the spermatogonia A. And another is spermatogonia B. Spermatogonia A and spermatogonia B. These are the two cells formed when the spermatogonia undergoes mitosis. So spermatogonia A and spermatogonia B. Okay, now what happens? What is there? Now spermatogonia A will again has the capability of dividing by mitosis and again give rise to A and spermatogonia B. So that is the nature of this cell called spermatogonia. Understand? Now then, therefore, what it can be called? Stem cell. It can be called as a stem cell. Keeps dividing, has the property of dividing and forming two groups again. So spermatogonia A again undergoes mitosis and keeps looping. Okay. So therefore here spermatogonia A and spermatogonia B. Spermatogonia A again further can form two. So this continues. So it has the property of stem cells of rapid multiplication. Now, therefore, finally, which cell can form sperm? Spermatogonia B can form the sperms. So therefore, here what they actually refer as, they refer to spermatogonia B. Okay. Now this what do you call this completes about explanation regarding the spermatogonia, from where did it come, okay, where it finally is present is within the seminiferous tubules. Of course, these all cells will be present inside the seminiferous tubule. All this process will be occurring inside seminiferous tubules. Inside the seminiferous tubules. And now, when this will start us from the time of puberty under hormonal influence. Otherwise, the cells will be, before the puberty, the cells will be in which is spermatogonia. They remain as only spermatogonia. Now, therefore, this stage, therefore, is called multiplication stage. This much portion is called multiplication phase. What is multiplying? Cells are multiplying and giving rise to two groups, the spermatogonia A and spermatogonia B. Multiplication phase. It is called multiplication phase owing to a term called mitosis, because the cells are multiplying. Understood? Okay, next. Next, what happens is the spermatogonia will start growing. So spermatogonia got split into the spermatogonia A and B, and this spermatogonia will start growing. So what is occurring is growth is occurring. So what happens during growth is it will start collecting the nourishing materials and starts increasing in the volume, or you can tell the size. The size of the cell is going to increase. It may vary from one organism to another. So here the size is going to increase. The spermatogonia, when the size is going to increase, means we refer to what is called the growth. And therefore, what is the cell formed after the growth? Only growth. No division is occurring. Only growth is occurring. That means spermatogonium B. B is undergoing growth to form primary spermatocyte. To form what is called the primary spermatocyte. Understand? This primary spermatocyte, since no division is occurring, the chromosome number since no division is occurring, the chromosome number remains the same. That means if it is 2n here, here also the primary spermatocyte will be 2n number of chromosome. That means here also it is 46. Here also for primary spermatocyte, the number of chromosome is 46. That is nothing but diploid, or nothing but 2n. Understand? So this was about the what is called here the spermatogonia will undergo growth. Okay. Now, therefore, from the next stage after multiplication phase will be the growth phase. How to identify? you can see spermatogonia will differ, and then what forms? Growth at forms. So here mitosis. So what you can do is, so here multiplication, and a very short phase is nothing but which phase? Growth phase. All the substance necessary, nourishing material necessary for further steps, that is meiosis, will be accumulated inside the cell, and as a result the cytoplasm can increase, and overall, not only cytoplasm, overall the cell size can increase, because at the, if you compare to the cell division during this time, interface is going to be there. Okay, so therefore here multiplication phase, and this will be, this part is going to be the growth phase. Now next is primary spermatocyte is the one that undergoes meiosis. So from here begins the actual maturation phase, it is called. So wherever the meiosis is occurring, that phase can be termed as the maturation phase. So maturation is due to meiosis here. So first maturation division, therefore it will be referred also. So next is primary spermatocyte is undergoing first meiotic division. Okay, first meiosis it undergoes. So meiosis 1. So here what? Primary spermatocyte undergoes meiosis 1. Now meiosis 1 is what? The chromosome number will become half in meiosis 1. They do not become half in meiosis 2. So therefore meiosis 1 will be called as reductional. Meiosis 1 is called as reductional, and therefore here only the chromosome number is going to get reduced into half. Okay, now this also has a name here. It is called first maturation division. It's called as first maturation division. It is called as the first maturation division. So primary spermatocyte undergoes meiosis 1. Now if I draw cell here, this particular cell, primary spermatocyte, now you should know that here the chromosome number is 2n. So till primary spermatocyte, and all cells before it are going to are diploid, and after that they're going to be, otherwise after the meiosis 1, after meiosis 1 they're going to be diploid. So this cell, this cell that undergoes this will undergo meiosis and produce 2 cells. Okay, these 2 cells. That means primary spermatocyte will undergo meiosis 1 to form 2 secondary spermatocyte. Both are capable of forming sperms, whereas it is different in case of female. So here this is called secondary, both are called as secondary spermatocyte. These are called as secondary spermatocyte. So secondary spermatocyte therefore will be haploid. Secondary spermatocyte will be haploid. As explained here, the meiosis is going to be reductional. So the chromosome number is going to be half, reduced to half here. 2n is going to become n. Understand? So 2n is going to become n here. Understand? So 2n, half of 2n is going to become n. half of 2n is going to become what is called n. Okay. So this is primary spermatocytes. Equal division will be there here, but in case of oogenesis, it is different. It won't be an equal division. That means the daughter cells have almost equal amount of the cytoplasm as well as, of course, equal chromosomes. So this is also called first maturation division, also called first meiosis. So therefore, next, because meiosis is occurring, which phase will start as maturation phase. Maturation phase is going to start. This is growth phase, multiplication phase, growth phase, and maturation phase. Okay, first meiosis and second meiosis. Now, secondary spermatocyte will undergo second meiotic division to form spermatids. So, if now I begin with secondary spermatocyte. For example, this is a secondary spermatocyte. This is undergoing meiosis 2. And it is not reductional. That is not reductional, but instead it is equational, because chromosome number is not changing here. It is equational. Chromosome number is equational. Understand? So since it is a second meiotic division, it is also called second maturation division. It is also called second maturation division. Got? Now this primary secondary spermatocyte will again give rise to two equal division is there. So two daughter cells, they are called as one secondary spermatocyte gives rise to two daughter cells, and they are called spermatids. They are called as spermatids. They are called as spermatids. Two spermatids. They are equal. What was the chromosome number here? N is equal to 23. Here it will not become half. It will not become n by 2. It will never become like that. It remains the same. It means that here also n is equal to 23. That is why justifying equational division here. N is equal to 23. Got? So this will be the second maturation division or second meiosis, by which cell? Secondary spermatocyte. And therefore, these are the broad stages in the spermatogenesis. Now, if you see this one, spermatids overall, if you see, this is the spermatogonia forming one primary spermatocyte, and primary spermatocyte forming two secondary spermatocyte, and each one again forming two what? Spermatids. If you see the number, one primary spermatocyte will give rise to 4 spermatids, and therefore we can now correlate with any of the number. Suppose it begins theoretically with 100, these will be 400. If it begins with say 200, further into 4. Okay. So if it is beginning with this one secondary spermatocyte is going to produce how many spermatids is going to be correlated with 2 into 2. So again can be extended, okay, to any other uh, values. Okay. Other values. Now what is formed is spermatids are formed. These 4 spermatids are formed. But these cells look like any other cells, very normal cells. They are not looking like sperms at all. So what is being formed is 4 spermatids have been formed, and these cell size is too big. The cytoplasm content is more. The organelles may be relatively less. The nucleus also is very large in size. It just resembles any other cell. But in terms of size and structure and all, it no more resembles the sperms and how sperms are formed. Now, these spermatids, what happens to the spermatids is they will start changing their structure, or you can tell morphology. Cellular morphology is changed there. Understand? So it means there is a morphological maturation, you can tell. It is not the physiological maturation. Okay, it's a structural maturation that is occurring here, where spermatids will start differentiating by a phenomena called cell differentiation occurs here. Remember, here also the cell difference is a very broad phenomena. Cell differentiation was occurring here also when spermatogonia differentiated into 2 and later on B underwent the growth. So here a cell differentiation is occurring here. It means that this particular spermatid is forming sperms. So spermatids now form spermatozoa. So what is spermatozoa? Group of sperms here. So you can, it's like another name of sperms. It is like another name of what? Sperms. So if you see here, it has a tail. The cell portion is too small, cytoplasm content, everything is too less here. But this spermatid has a lot of all these contents, what I mentioned before. So now therefore this cell has to undergo differentiation, that is called cell differentiation, and then it should form what? Sperms. Now what is this phenomenon undergone here? Cell differentiation. It has to undergo cell differentiation. This cell differentiation has particular term used, that is called spermatogenesis. It also has many other, it also has other terms like spermatolysis. So this is what, students? This is spermatids, and then it is undergoing differentiation to form sperms. Understand? The term here used is spermatogenesis. So what is occurring here is spermatogenesis is occurring. Understand? So here at this point, the phenomena that is undergone is spermatogenesis. So what is spermatogenesis? Spermatogenesis is where a phenomena where spermatids will mature into sperm. What happens here? It is not active. The sperm is active. It is non-functional. This is functional. It means not totally functional, but it will start gaining more functions here. Okay. It is non-motile, and it becomes motile here. Non-motile means not, I mean, motile means ability to move here. So basically, spermatid is non-motile, but the sperm is going to be motile. Now, how this differentiation is occurred is due to spermatogenesis. Okay, that differentiation is going to occur. Okay, so therefore here what occurs is spermatogenesis occurs. Process where spermatids excuse me process where spermatids mature and form what? Sperms or spermatozoa. Sperms or spermatozoa. And this whole process itself is called spermatogenesis. Whole process is called as spermatogenesis. And spermatogenesis is nothing but cell differentiation. Spermatogenesis is nothing but the cell differentiation. So maturation continues here. Okay. So this term which you should know here is spermatogenesis. What all changes can occur here? Spermatid is quite bigger in size. So cell size reduction occurs. That means how is the cell size reduction occurs? Not because of reduction of the chromosomes cannot be deleted, but uh, what is reduced is the cytoplasm content is reduced. When cytoplasm content is reduced, then the cell size also becomes lesser and lesser. The cytoplasm becomes so less that it just remains as a rim when in the sperm. Okay, a thin layer of cytoplasm is formed in the sperm because of cytoplasmic reduction. And the second is the nucleus gets reduced in the size by condensation. So nucleus is not getting reduced due to removal of chromosome and all. It is actually getting reduced due to condensation. Okay, so certain substances are removed other than the chromosomes. So condensation occurs, which you have studied in the cell division itself last year. So that is ultimately contributing to reduction in the size of the nucleus. And the Golgi bodies are able to produce acrosome during this spermatogenesis. And you'll know that acrosome is very important for the sperm to penetrate into the ovum. And second is the mitochondria will further divide. Duplication of the organelles occur. The organelles can, like mitochondria, and that then what happens? It will start surrounding the microtubular structure in the midboard, in the middle region of the sperm. Whereas the structures like microtubules, they will organize themselves starting from the centriole. They will organize and form the tail axial filament, which will run till the tip of the tail. And therefore, during this differentiation, also tail of the sperm is formed, and microtubule is one of the important components within the tail. And it is a structure responsible for motility of the sperms. Even that is formed during this spermatogenesis. When the cytoplasm is excluded or resulting in its reduction, that excluded cytoplasm can be absorbed by the surrounding Sertoli cells. You'll know that seminiferous tubules, and say this is the Sertoli cells, only one side I'm drawing here. So as this say spermatids forms the sperm here, its cytoplasm, excluded cytoplasm can be absorbed by these cells called as sustentacular cell, also you can call it as nurse cells, or also called as Sertoli cells. So remaining of the spermatogenesis, I'm going to explain in the next class.